Hi, everyone. Welcome to part 2 of biological macromolecules. Today, we're going to talk about proteins and nucleic acids. But first, we're going to talk about nucleic acids. So remember how in the first video, we talked about the fact that all living organisms need to use information to actually grow and reproduce and kind of use the data within like their genes and like a kind of figure out what's happening around them, right? So the way they do this is using DNA and RNA to actually store this information. And what do these RNA and DNA or these nucleic acids look like? These are nucleic acid polymers where DNA is double-stranded and RNA is single-stranded, like you see here. And in general, they are made up of nucleotide monomers. A nucleotide itself has three components: a phosphate group, a 5-carbon sugar or a pentose sugar, and a nitrogenous base. If you guys don't remember anything from this class afterwards, remember this part here, because it is crucial to understand how our genes are set up, right? And these involve these are your basic units that make up who you are. And in this case, the sugar molecule is carbon, fifth carbon. So this one is connected to the phosphate, and the first carbon of the sugar is connected to the nitrogenous base. Now, one thing that you need to know is that the RNA and DNA sugars are completely different, or not entirely different. It's one aspect that is missing. It's so in RNA or ribonucleic acids, the sugar itself is ribose. So this is ribose with the OH at the 2' carbon. But then when we look at DNA or deoxyribonucleotides, the sugar itself is called deoxyribose. And that is because it is lacking the oxygen in the 2' carbon where we would see it in RNA. Now, the main difference is that single oxygen atom that is missing, and all of the 3' carbons have that hydroxyl group. Now, the nitrogenous bases within DNA and RNA have are in two groups. One is called purines, which have these bigger structures. The pyrimidines are these smaller ones here. And what the two that are purines are adenine and guanine. And then the pyrimidines are cytosine and thymine in DNA. And then cytosine and uracil in RNA. And when these nucleotides come together and polymerize, that's when you have nucleic acids. These are basically polymerizing to form that RNA or DNA, depending on which nucleotides you have. And these are linked together by bonds called phosphodiester bonds. These are, again, formed through condensation reactions where they end up removing a water molecule. So now what we have is the DNA and RNA having one end that is unlinked, 5' here, and then the other end here has the 3' that goes unlinked. The sequence itself is written and read in the 5' to 3' direction. But when it adds on the nucleotides, it adds on to the 3' end. And the sequence itself is the primary structure of DNA or RNA, depending on which nitrogenous base you have, which sugar molecule you have, so on. Now, way back in the day, like 1950s, there was a scientist named Erwin Chargaff. He was looking at the molecular structure of DNA, and he basically established two empirical rules for DNA. And this kind of later became known as Chargaff's rule for DNA itself. And what we see is in a DNA sample from an organism, the total number of purines is equal to the total number of pyrimidines. So it would be like A's and G's equal to C's and T's. And another part of his rule was that in a DNA sample from an organism, what happens is that the number of adenines and thymines are equal. So, like A's and T's are equal, and the number of C's and G's are equal. So, if we have like a question where we ask a bacterial species has 20% thymine, how much do we have of adenine, cytosine, guanine? So from Chargaff's rule, what we know is that we have 20% thymine means 20% of adenine, and if we add that together, it's 40%. What happens is that this all of this adds up to 100, where if you take out the 40 out of the 100, that's 60, and then 60 divided by 2 is 30. So C would be your answer there. So now a couple of years later, people were interested, like, ooh, what's happening here? Let's figure out how the DNA is actually structured. And in 1952, 1953, around that time, Rosalind Franklin, Maurice Wilkins, James Watson, and Francis Crick ended up determining the structure of the double helix. And what they saw there was there were two anti-parallel strains, which means that one was going into from 5' to 3', and then in the other direction it went from 5' to 3'. And these were the hydrophilic sugar phosphate backbones were at the edges, and the interior is where all the nitrogenous bases were paired, and it had certain number of hydrogen bonds to keep them stable with pairing each other. And what was seen is that adenine paired with thymine with 2 hydrogen bonds, and then with guanine and cytosine, there were 3 bonds together. And what they realized was that the double helix itself is actually the secondary structure of the DNA, sorry. The double helix is the secondary structure for DNA, and we see that with these structures shown here. So now we're going to look at more of the characteristics of how RNA is structured. So the primary structure itself differs from DNA by the presence of uracil instead of thymine, and the sugar is ribose instead of deoxyribose, and the hydroxyls on the ribose make RNA more reactive, and a lot less stable. So these are causing it to be more reactive. It can also catalyze different chemical reactions. These RNAs that are catalysts are called ribozymes. And biologists tend to think that these RNA existed way before DNA was involved. So now the secondary structure, they are single-stranded, and the structure itself results from complementary base pairing when it folds over each other within that same strand itself. Sometimes they just fold over, forming like this hairpin loop, and this anti-parallel segment ends up folding over each other like a little loop. The unpaired bases end up forming a loop, and then the RNA strand ends up forming this double helix kind of areas where the two regions can actually do a base pair. So now the tertiary structure of RNA is basically where the secondary structures kind of fold into a more 3D complex shape. For example, like a transfer RNA molecule. We'll talk about those more in a bit. And the RNA itself can function as an information containing molecule, but it also carries out catalytic and regulatory functions. And there are certain catalytic or enzyme like RNAs that are called ribozymes that can actually boost and speed up chemical reactions. And for functions of RNA, those are more in terms of like protein synthesis. Those involve three different types of RNA: messenger RNA, that contains the instructions to build these proteins, transfer RNAs that help with the protein synthesis. Same goes with ribosomal RNA. So they kind of are in the nucleus, migrates into the ribosomes, and kind of the rest of them are in the cytoplasm, cytoplasm. And these provide linkages between mRNA and amino acids for the transfer RNAs, and the ribosomal RNA are more structural components. And then, like we talked about, there are catalytic ones and regulation of gene expression, where these are functional RNAs, where a certain types are like ribozymes or microRNAs. And with that, we go into our last macromolecule, which are proteins. So proteins are basically crucial to anything to occur in a cell and allows them to exist properly. Many of these functions include catalysis, defense mechanisms, signaling pathways, movement, structure, transport, fluid balance, gene regulation, so on. And when we look at most of these, it ends up being critical in how these are regulated. These proteins that are involved in these are also very crucial to what is actually seen. And these are just some examples that you guys should know. It comes in handy in understanding these different functions of proteins. So what do these proteins actually look like? These structures are very, very diverse. like these here. And like we talked about with carbohydrates, structure determines function in this case. So like if we have like certain cases where it's like saddle shaped like this, it could bind to DNA. If it's donut shaped, it forms a pore where things can go through. If we have like a globular kind of shape, it could be like active site, uh, the active site that binds different substrates for enzymes. And then if it's like fibrous like this, it provides structural support. So depending on the structure, it can tell you what the function is. And so what are these proteins actually made of? When we look at proteins, these are polymers of components called amino acids, and these are linked by peptide bonds. There are specifically 20 naturally occurring amino acids, and they each have the same general structure, where it's a central carbon, a hydrogen group, an amino group, the carboxyl group, and the side R chain that is distinctive among the different amino acids. So amino acids, when they are in solutions, they can ionize in like body fluids or water. Typically when they're in water, it ends up ionizing to these groups respectively. And what happens is the non-ionized form is like this, but then when it's in like a liquid in a aqueous solution or like body fluids, what happens is that you have ionized amino acids. And this essentially helps amino acids stay in the solution and makes them more reactive with the other molecules around. So when we look at the biochemical characteristics of the side chains, these side chains are what makes them distinct from each other and how they end up affecting the properties of the proteins themselves. And these amino acid side chains can be grouped into 4 general types, including acidic and basic, which are electrically charged. Then there's uncharged polar and then non-polar side groups. So when you look at these amino acids, there are 20 amino acids like we've talked about. Each of them has a different R group, and depending on the R group, we see the difference in the chemical nature of each amino acid, depending on how they look, how they react. You do not need to know the structures of these different amino acids. Just keep in mind that some of them can be nonpolar, some can be polar, others are like nonpolar, negatively charged, positively charged. Knowing the main groups of these amino acids are helpful in understanding what properties exist for them. So now what we see is another condensation reaction, which basically brings the carboxyl group of one amino acid and the amino group of another amino acid, bring them together, take out the water molecule, and you have a peptide bond. And that is what you see here. And this polypeptide is basically a chain of amino acids linked by many peptide bonds. So polymerized amino acids are called polypeptides. When you look at these here, they are all linked. They have different side chains, and this N terminus and C terminus. It's the beginning towards the end of the protein. Okay, so if we see the N terminus or the amino group terminus, that's your beginning of the protein, and then the C terminus or the carboxyl terminus is the end of that protein. Now, like we all say, structure determines function. It's a critical aspect of what is happening in our cells, and we will talk about in a little bit how if that changes, it causes chaos, essentially. So what happens is proteins have 4 basic levels of structure, first being the primary, secondary, tertiary, and quaternary. So the first structure itself is basically the unique sequence of amino acids that are not forming any other structure other than the sequence itself. This is basically the information from the gene that is encoded for that protein. And there's 20 types that can be used and can be used in many different combinations. And it can go from 2 amino acids to tens of thousands in a protein set. Then we have the secondary structure. The secondary structure is formed by hydrogen bonds between groups in the backbone of the amino acid, not the R groups. So specifically for like the amino groups and the carboxyl carbonyl groups, what happens is that these occur, and what happens is that they form 2 specific shapes. One set would be the alpha helices, and the other one would be the beta pleated sheets, like these. Alpha helices, beta pleated sheets. Now, another set to this is the tertiary structure, where it starts making interactions between the different R groups. So secondary structure was basically through the backbone structures. Now we're having interactions between R groups of the structure itself. And what we see is that in these type of R group interactions, we have a couple of different bonds and interactions involved in protein folding, including hydrogen bonds, hydrophobic interactions, van der Waals interactions, and covalent disulfide bonds and ionic bonds. Now it takes it even another step further and starts bringing in different polypeptide subunits that basically combine to kind of form a single structure, and basically brings in the quaternary structure. So like hemoglobin tetramer, so like we have like alpha 1, alpha 2, beta 1, beta 2, and these all come together to make one hemoglobin tetramer. So now, if we have issues in protein folding, we are completely hopeless, because in some cases with these, if they are not folded properly, they won't be functional. Like we said, function is determined by structure. So if it's not structured properly, it will not work properly. So when we look at these, it's like it's often spontaneous because it's very energetically stable in folding the molecule properly. So we have some proteins are actually there to help proteins fold correctly within the cells, and these proteins are called molecular chaperones. They're like, okay, people, get it together. We need to move around and keep it all together and fold correctly, or we're gonna be having nightmares for like million years. And what happens is that if they are not folded correctly, it can cause diseases, and like especially if they're not degraded from the system, it ends up causing many diseases. And in some cases with like a these unfolded or denatured proteins, what happens is they can't function normally, but it can be a reversible process depending on the nature of how it was denatured. So if it's like a change in the salt concentration or pH, it can denature or renature the protein structure, but other times it's not reversible. It can be like this poor little egg where it's like all folded and correctly, but then when you cook it, it's all denatured and you have no more um proteins that are uh not um disrupted. Similar to like other, so this is heat related denaturation. Then there are other types where it's like heating, radiation. Sometimes it's like whipping up some cream here, and so on. So that kind of brings us to the end of our biological macromolecules unit. Next, we'll go into the uh, cell structure and function chapter. Until then,